Hello friends, this video on molecular basis of inheritance part 30 is brought to you by examfear.com. No more fear from exam. So now we will talk about regulation of gene expression. So we saw that the genes are the genetic information as being stored in the DNA. When we talk about expression of the genetic information, then we talk about RNA. That is, we talk about transcription and translation. So finally, when translation happens, the genes actually get expressed. So here we will talk about how can we control the expression of genes. So let us see what do we learn out of this topic. So before we talk about regulation of gene expression, let us first try to see what is gene expression. Now, as I said, the process by which genetic code directs protein synthesis, that is gene expression, because protein synthesis is the process where exactly the genetic information which was stored in the DNA is getting expressed in the form of the traits present in an individual. So that is the meaning of gene expression. So when we talk of gene expression, there are two processes which are part of gene expression, that is transcription and translation. So in transcription, what happens is the information which was stored in DNA helps to form the RNA, and RNA which is formed, that can directly control the protein synthesis. So the RNA then can be converted into proteins by the process of translation. So this is how the genetic information that was stored in DNA gets expressed in the form of traits. So here in this picture, you can actually see this entire portion is the nucleus, and inside the nucleus you have the DNA. So the DNA duplicates, then the DNA undergoes transcription. So here you can see the process of transcription when RNA polymerase enters beyond the promoter region, and then it elongates the transcription unit or the structural genes of the DNA, and then an RNA is formed, and this is the mature RNA. So in order to convert the RNA into mature mRNA, several processes like RNA splicing takes place, and this mRNA, that is the mature RNA, finally comes out of the nucleus into the cytoplasm, and this is where you can see the mRNA. Now here you have the ribosome. So the mRNA reaches near ribosome, and meanwhile the tRNAs, they carry amino acids, and depending, they can read the sequence on mRNA. So reading the sequence on mRNA, they provide the corresponding amino acid, and these amino acids are then joined together by peptide bonds, and this is how proteins are synthesized. So this is the overall process of transcription and translation. So this is how genes are expressed. So what do we mean when we say regulation of gene expression? So this is nothing but controlling the gene expression to ensure that appropriate genes get expressed at the right times. Now there can be scenarios when we do not want the genes to get expressed. It is not necessary that we always want all the genes to get expressed. Sometimes we might not want few genes to get expressed. So that control on the gene expression, so that the right genes are expressed at the right times. So as I said, not all genes get expressed all the time. So let us take some examples. Now let us suppose there are genes which control the synthesis of proteins, and those proteins participate in the basic metabolic processes of our body. Now those basic metabolic processes in the body will be taking place continuously. So we want such genes to be expressed continuously. We want them to be expressed at all times. However, if we have another set of genes which express for proteins or which code for proteins which help in embryo formation, that is growth and development of the embryo. Now growth and development of an embryo does not take place all the time. So these kind of genes can be expressed only at the right times, because at other times we do not really need them, because we do not really need those proteins. So we can just ask those genes not to synthesize proteins. Now how can we ask the genes whether to synthesize protein or not? So if the genes, it is like a switch. If the genes are turned on, they will synthesize proteins. If the genes are turned off, they will not synthesize proteins. So this control for switching on or switching off the gene is known as regulation of gene expression. So now there are various ways, or there are various stages where the gene expression can be regulated, because we already saw how gene expression takes place. So now where exactly can we control the way gene gets expressed? So gene expression can be regulated at any of these stages, like transcription. For example, the rate of transcription, the rate at which transcription happens, that can be made faster or slower. Processing of pre-mRNA to mature mRNA. There are quite a few processes which takes place for converting the initial mRNA into mature mRNA. For example, capping, tailing, RNA splicing. So all these processes take place. So there also the gene expression can be controlled during any of those processes. Transport of mRNA from nucleus to cytoplasm. Now when this mature RNA gets transported from nucleus to the cytoplasm, there also the gene expression can be regulated. So these are some of the stages where the gene expression can be controlled. Now let us see how exactly the gene regulation or the control is actually done. So it can also happen during translation, as I said. Now transcription and translation, that is the place where gene expression actually takes place. So control can also be done at that time. Now, just now I was talking about the examples that the proteins which help in the basic metabolic activities, they need to be synthesized continuously, whereas those proteins which are responsible for growth and development, they might not be needed all the time, because you would have seen that the growth and development of certain parts of our body stops after a certain age. So in that case, we do not need those proteins anymore. So the gene expression can be controlled for proper expression at proper times. So for the first time, this Operon model to explain the gene expression regulation were explained by a group of 3 scientists, Loew, Jacob and Monod. They developed this concept of Operon model to explain the regulatory mechanism in prokaryotes. Now what is this Operon model all about? Now the term Operon, this term Operon means to operate. So how something operates, that is what it means. So the story all started with bacteria E. coli and viruses. So it was found that there was a mechanism to turn off or turn on a set of genes depending on the requirements. So if a particular set of genes was required to synthesize a protein, they were turned on. If the protein synthesis for those genes were not required, then the genes were turned off. So there was a mechanism. So whenever the genes were turned on, proteins were synthesized. Whenever the genes were turned off, proteins were not synthesized. So these 3 scientists were awarded Nobel Prize in 1965 for their work on the Operon model. So now we will spend some time trying to understand the functioning and the working of the Operon model in case of bacteria. Now there are various Operon models which acts for different purposes. It is not that there is just one Operon model acting in a bacteria. Several Operon model works. So we will just discuss one Operon model just to get an idea about how the entire concept works. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.